estimate what proportion of all GE organisms would be exempt and non-notifiable? If you can't, can you recommend a scientist in New Zealand that has evaluated this and could understand this?
We're not aware of anyone who's evaluated this. The point is that exempt activities, which is a weird way of saying particular products, and non-notifiable activities, which is a weird way of saying the process by which you make these things, would be outside of regulatory overview. And that means that you don't control what organisms are exposed to the genome editors, and you don't control who exposes them to the genome editors. You can use extremely common and areas in your home or around a school or on your lawn. None of these would be regulated, and by people who aren't technical experts in the field. And I don't say that to be dismissive, because technical experts can do pretty stupid things too. But at least under current regulations, even if a technical expert does something stupid, it's inside of a contained facility. And all the stupid things can be rendered inert and not released inadvertently, hopefully, into the environment. That would change. So the proportion of organisms that are altered using these exempt processes would be astronomical. To give you some sense, if I do this outside, I'll be exposing bacteria. There are billions of bacteria per gram of soil. There are hundreds of millions of bacteria per cubic meter of air. All of these would be simultaneously exposed. And it's a fallacy to think that we know the genomes of all these organisms, and that the tools that we use to direct those mutagens to particular DNA sequences in their genome, that those tools will exclude sequences that are in the genomes of all those billions of different species. Our databases, even if they could tell us these things by being complete, are not complete. On top of that, all of our cells have latent viruses, and some of our cells may be under an active viral load from an infection, even if it doesn't result in symptoms. Those viruses comprise a virome for which we have almost no database information at all. And that means inadvertently, any of these tools used at any time could be making changes in organisms that we have no intention of changing. The vast majority of those changes will probably be irrelevant from a biosafety point of view, but at the scale we're doing it, and the failure to record what organisms we're doing it in, and the inability to contain them and destroy them before they're in the environment, that combination of new variables we cannot dismiss as negligible risk.
And there is in the scientific record, there is a record of inadvertent alteration of not just the DNA they thought they were working on, but DNA that was next to the cell, for example.
Well, and I would just point out that even in the intended organism, you can't control all the unintended changes. The use of the word precision by practitioners is really misleading in my view. These reactions are highly efficient, but we do not completely control the location in the genome at which they are being efficient. But even beyond that, there's a problem, and that is the reagents we use, the specific ingredients in the reactions. These come from biological sources. And each of those biological sources is contaminated with DNA from the pipeline of production. So even if you're intending to only use a type of process that creates an exempt activity, an activity where the product has only had a site-directed nuclease damage the DNA, and that damage should have been repaired using processes that are endogenous to the cell, and therefore not producing a new sequence from another species, for example. Even if that's your intention, the you cannot rely on that being the outcome because there's contaminating DNA in your reagent line and potentially just from the contamination of the where you're doing it. And if it's not in a contained laboratory, there's even more contaminating DNA sources. So amongst the mix of products that you invariably make every time you use a gene technology will be a substantial number of those that we traditionally call transgenic organisms, and which this bill continues to regulate in higher risk tiers. The exempt activities cannot be considered or assumed to be exempt activities until they're verified and proven to meet the exemption criteria.